Hello, today I'm going to talk about fatty liver. Fatty liver is status when fat concentration of total liver is more than 5%. It is categorized whether it is occurred by alcohol or not. Former one is alcoholic fatty liver disease. Later one is NAFLD, non-alcoholic fatty liver disease. Fatty liver keeps in progress. It leads to steatohepatitis, SH, which involves fibrosis or inflammation, liver cell damage, and if it worsens, it leads to liver tumor. This disease is highly related to glucose fat metabolism by insulin action. Liver is the organ that detoxifies toxic waste. Drinking alcohol detoxifies in liver by enzyme ADH. ADH produces NADH, which consumes NAD+. It makes beta oxidation of fat harder. Alcohol finally produces acetyl-CoA, which is used to make fatty acid. Gene mutant can occur NAFLD. Patatin-like phospholipase domain containing protein involved in glycerolipid hydrolysis, which is highly expressed in liver. C2G mutation promotes fibrosis in liver inflammation. Also, PNPLA3 degradation avoids, which damages triglyceride move in lipid drumlet. TM6SF2 mutation reduces pre-LDL triglyceride secretion related to fatty liver, and this mutation reduces cardiovascular event. MBO87C2T mutation changes lysophosphatidylinositol acetyltransferase, which control arachidonic acid to synthesize eicosanoid. Prostaglandin is kind of eicosanoid. So when this gene mutated, blood lysophosphatidylinositol increase act as GPR55, that is kind of GPCR ligand, and further signal transduction promote liver fibrosis and fat synthesis. Other gene involved in fat metabolism, glucokinase regulator, insulin signal transduction. How NAFLD formed? De novo lipogenesis occurs in liver tissue. DNL is the process to convert carbohydrate to fat. In the process, fatty acyl-CoA is used to synthesize triglyceride. This substrate concentration is determined by several factors including free fatty acid, lipogenesis, lipoprotein absorption, TG breakdown, lipid oxidation balance. When each homeostasis breaks, fatty liver occurs. Composition of liver triglyceride in NAFLD patient was non-esterified fatty acid, NEFA, 59%, which is bound to blood albumin to flow in blood environment. So which free fatty acid in blood. Fatty acid from DNL involves in NEPA pool, and NEPA fat can store back to liver. NEPA contributes mostly to fatty liver. This picture is about insulin receptor signal pathway. When insulin binds to insulin receptor, it is kind of tyrosine kinase, so it self-phosphorylates tyrosine residue. It promotes docking of substrate IRS1, IRS2. PI3K docks to phosphorylated IRS1 or IRS2. It converts PIP2 to PIP3, and PIP3 recruit PDK1 is kind of kinase. PDK1 phosphorylate AKT. AKT phosphorylate two transcription factor. One is FOXO1, reducing gene transcription involving genetic space. PepCK contributes to glucose synthesis. Also, AKT activates SREBP1C, steroregulatory element binding protein, acetyl-CoA carboxylase, fatty acid synthase, ATP citrate lyase upregulates. Those enzyme amount is increased, and fatty acid biosynthesis activates. Many factors in this picture and further slide disrupt this pathway. But the insulin is well known as accumulate fat. How disrupting this insulin pathway affects to produce accumulate more fat. Fat accumulation lead inflammatory cytokine, ER stress, oxidative stress. Those factors promote JNK, that is kind of kinase. JNK overactivation and inhibit IRS2 by phosphorylate serine residue. RST reduces increased insulin resistance. Also, DAG accumulation inhibit insulin receptor. How insulin resistance increase liver fatogenesis? First, in the DNL pathway, gluconeogenesis inhibition and lipogenesis activation pathway are divided. Many factors affecting this pathway, such as diacylglycerol, only break gluconeogenesis pathway, so lipogenesis is conserved. Second, insulin-independent pathways such as the insulin-independent ChREBP reacts to glucose intake. Gene involved in lipogenesis turned on independently. Obesity and high amount diet leads to fatty liver regardless of insulin by ChREBP. Third, IRS1, IRS2 signal molecule expression differs in liver. Hepatic portal vein surroundings, IRS2 expression is bigger, but in hepatic vein, IRS1 expression is bigger. Fatty liver patient blood insulin is high, so insulin pathway reinforce IRS1 surrounding hepatic vein, increase DNR. Fourth, ER stress involves in insulin resistance, but it also activates SREBP through independent pathway, increasing DNR. How NAFLD cause symptoms to patient? Fat excess, high fat diet cause excess TCA cycle activity, so oxidative stress increased. And mitochondria maximum respiration rate observed bigger in obesity. Mitochondria damaged or leak out, which cause more oxidative stress and inflammation, further hepatocyte damaged. CD36, kind of glycoprotein involved in free fatty acid absorption and movement in cell. Overexpressed in liver cell, increased triglyceride storage related to fatty liver. Normal adipose tissue formed by angiogenesis, which is blood vessel formation, and ACM remodeling and fat engages. Pathologic adipose tissue enlarge basal adipocyte, but angiogenesis is limited for enlarged tissue. This causes hypoxia, which is lack of oxygen, to adipocyte. Hypoxia-inducible factor activates HIF-1 alpha in hypoxia environment, which activates gene transcription for fibrosis, insulin resistance, and inflammatory phagocyte activation. Insulin prevents fat breakdown. Insulin-resistant tissue release free fatty acid. This circulatory fatty acid activates NF-kappa beta pathway in liver, which accumulates triglyceride-originated toxic metabolite in liver. Previous slide, we talked that abnormal fat increases inflammatory phagocyte. When it happens, inflammatory cytokine tumor necrosis factor TNF-alpha is upregulated, which promotes adipocyte breakage, insulin receptor serine residue phosphorylation, and it inhibits tyrosine phosphorylation. This is kind of insulin resistance. This process increases insulin resistance in the whole body. Many patients in this state has higher interleukin-6 in their blood. Interleukin-6 increase can also contribute to insulin resistance by breakdown of insulin signal pathway molecule. Adipocyte also secretes specialized adipokine. Leptin has appetite inhibition function, but obese people has leptin resistance. Leptin circulates blood is higher in obesity. This state promotes TGF-beta signaling to promote fibrosis. Also, toxin reactivity increased because of leptin resistance. Adiponectin has ADIPR1 and ADIPO-R2 receptor. It involves in positive pathway, activates AMPK, activates PPAR alpha, increase insulin sensitivity. But obesity has lower adiponectin due to oxidative stress, so fat is increased. Skeletal muscle use insulin-dependent glucose transporter 4-mediated glucose transport to control blood glucose level. Due to excess free fatty acid supply, fat in muscle cell increase, which interrupts DAG-mediated insulin signal transduction and promoting insulin resistance. This means glucose uptake is not normal in muscle, so it also could be used to de novo lipogenesis. This process is referred to symptoms such as muscle reduces, muscular strength reduces. Gut microbiome reacts to change of diet, which can lead to unbalance. Unbalance could disrupt gut endothelial cell tight junction. Cell permeability increased, and problem is when gram-negative bacteria move into gut epithelial cell. It contains lipopolysaccharide, which act as endotoxin. Fat tissue inflammation, liver triglyceride increases. and small intestine bacteria overgrowth provoke liver damage due to endotoxin flows through hepatic portal vein. Also, bacterial endogenous ethanol synthesis could contribute to fatty liver. Viral fat could result from hepatitis virus HCV. It is topologically unable to distinguish from fatty liver, and it could further lead to fibrosis HCV. Many surveillance data shows HCV patient is susceptible to IR, which may lead to type 2 diabetes. Glucose goes through glycolysis, and some steps are spontaneous. To maintain homeostasis, those steps are regulated by activator and inhibitor. When fructose are used for energy source, they have low affinity to burst enzyme of glycolysis. So fructose are oxidized by fructokinase at first, and further step occurs. Aldolase B in liver directly cleave fructose phosphate to 2,3-sugar, which converted to G3P, and it involved in glycolysis uh, second chain. So regulation step skipped when fructose are digesting. This makes fatty acid precursor with no regulation. Excess fat could be produced. This is summary of fatty liver cause. Fatty liver doesn't appear much symptom, but when it is progressed to inflammation, it may cause fatigue, light blank ache. Metabolic syndrome and fatty liver are bidirectionally cause and effect, so metabolic syndrome could be symptom. Liver damage could lead to hepatocyte enzyme ALT increase, blood insulin, LDL increase. Now it is therapy. Hepatokine is protein that liver produces. It interacts with adipose tissue, muscle, immune system, and etc. Bad hepatokine such as betaine A binds to insulin receptor and cause insulin resistance. Such mechanism could use to make drug. There are also good hepatokine. Fatty liver changes hepatokine secretion type. Many drugs inhibiting bad hepatokine and activating good hepatokine are in developing. For example, phase 2b drug proxyperamine increased FGF2 signal and increased insulin sensitivity. Metformin activates AMPK to inhibit SREBP protein and increase beta oxidation. Pioglitazone is agonist to target PPAR gamma to increase fatty acid oxidation. Glucagon-like protein 1 can indirectly activate PI3K-AKT pathway to reduce fat accumulation. Borzocostat inhibit DNR. Probiotics may be helpful to improve gut microbiome. Bariatric surgery that detour or removing stomach rapidly recover liver inflammation. Losing weight positively affect because adipose tissue reduces lead to liver fat reduce. Weight training also increase muscle insulin sensitivity. Quit alcohol, fructose is helpful to prevent fat production. Thanks.